Hello and a very good day to all Biotechnology competition participants and honorable judges. I hope this speech finds you well and safe. I'm Lim Si Jie, a PhD candidate from Universiti Putra, Malaysia. I'm very glad to participate in this Biotechnology competition organized by the Faculty of Arts and Science from the International University of Malaya, Wales, with the theme Biotechnology is the future, moving forward from today. In this ever-improving field of biotechnology, many aspects relating to biotechnology have been progressing very quickly over the years. This field is a broad discipline where biological processes, organisms, cells, or even cellular components are exploited to develop new technologies and products. However, one should always remember that the progress in biotechnology is closely related to the industry which generate revenues from achievement in the modern biotechnology. Having said that, proteins or enzymes play important roles in the industries. These enzymes can speed up the process in industry, such as amylase, to convert starch into high fructose content syrup. The industry important enzymes such as lipases and amylases have been widely used in food, detergent, and many other industries, where they boost up the function of the dishwashing detergent by breaking down the lipids and also carbohydrates on the dishes. Ladies and gentlemen, do you know how super enzymes work? Basically, these enzymes are very unique in terms of their 3D structure. Here, I would like to share with you a little bit on the structural biology and how it impacts on the expression in industrial field. Most people may think that it is just the structure of a biomolecule when it comes to structural biology. However, in nature research, it is defined as the study of a molecular structure or dynamics of a biological macromolecule, particularly in proteins and nucleic acids, and how the alteration in their structure can affect their functions. My fellow friends and outstanding researchers, considering the huge and important information can be obtained from protein structure, the next question will be, how do we solve these 3D structures? Yes, in structural biology research, the tertiary structure of a protein can be elucidated using nuclear magnetic resonance (NMR), cryo-electron microscopy (cryo-EM), and also X-ray crystallography. Wait, it may sound really complicated and difficult, right? But no worries, there's an alternative and easier way to do it. The protein structures can be predicted via homology modeling or protein trading platforms through the bioinformatics databases and tools. Guess what? Using all these in silico analysis and platforms, the new enzymes with enhanced features can also be designed and engineered towards the industrial desired traits by predicting the critical points of mutations. The interactions between amino acids, residues, and the secondary structures can be clarified, such as the hydrogen bonds, salt bridges, and many others, where they can be controlled by adjusting the amino acids and also the condition of the protein itself. It is interesting to know that a recently published paper by Chan et al. In Science described the deep mutation in human angiotensin converting enzymes to ACE2, which could increase its affinity towards a spike protein of the SARS coronavirus 2 and neutralizing it. What an amazing milestone in the medical field, right? Ladies and gentlemen, I believe this speech has stimulated a little bit of your curiosity towards structural biology. Now, let's move on to the final section of this talk. When it comes to the protein expression in the industries, the microbial expression host with the highest recombinant titer will always be preferred. Bulk production of the proteins in industries can be induced in a bioreactor and subsequently separated, purified, and spray-dried for further usages. To conclude, understanding the structure and keep the enzyme at its native state are important, but modifying the enzymes via protein engineering could be very helpful and significant for its high industrial demands. Hence, biotechnology is the future where the trend has always been progressing. I hope what I'm sharing today can help generate new ideas, especially in structural-based protein engineering, so that a novel industrial important enzymes can be successfully designed and produced. Thank you very much.